He is currently an associate professor of medicine and biomedical engineering at Washington University School of Medicine. He received his PhD from MIT and completed his postdoctoral training in the laboratory of Dr. Douglas Melton at Harvard University. His current research is focused on synthesizing both biomedical engineering and cell biology approaches to use stem cells for the study of and treatment of diabetes. His innovations have been licensed to several biotech and startup companies in the diabetes space and resulted in one ongoing clinical trial. His laboratory is supported by NIH, NINDDK, and JDRF, and his scholarship has been recently recognized by awards from JDRF, American Institute of Chemical Engineering, and the Biomedical Engineering Society. Without further ado, the floor is yours, Professor. You're muted again.
There we go.
Okay.
Yeah, just trying to get it all set up on my side. Too many buttons hit at the same time. Okay, thank you very much for that introduction. I'm going to move the camera so I can actually look at what I'm pointing at here. I really appreciate the invite. I haven't been able to, as we we're just talking about before getting onto the presentation here, I haven't had a chance to interact much with the Institute previously. So I'm glad to be able to at least take an hour to share my work with you all to begin to engage more with your institute. So I really appreciate the invitation to be here. I'm going to start off by first saying basically what the topics are today. It's going to be a pretty broad presentation in terms of topics, but it's all going to be focused on making producing beta cells and islet cells for the study and treatment of diabetes. So I'm going to go into the motivation, how do we do this, what can we do with these cells, and a kind of forward perspective of kind of what I think that the future is going to hold, and some maybe case studies in terms of how that could potentially look like. Before really getting into the meat of this presentation, I wanted to acknowledge my team here at Washington University School of Medicine. So I'm a chemical engineer turned stem cell biologist. And the composition of people in my laboratory reflects that I have people from either the medical campus's umbrella PhD program, one MD PhD student, and I also have several students who either have PhDs in bioengineering or are in the bioengineering PhD program. And I think that's a good reflection of the type of research that we do in the lab, where we try to be very translationally focused, and I think having a good balance of biology backgrounds and bioengineering backgrounds is critical, at least the way that I approach it, to be able to be successful in translational research. To start off here, I have a few slides just kind of summarizing in very broad strokes the disease of diabetes. And there's a lot of different forms of diabetes, but essentially diabetes boils down to there not being enough healthy beta cells within the islets of the Langerhans, which are located within the pancreas, in order to properly control blood glucose levels in patients. And this could be because the patient just needs a lot more insulin than the beta cells are able to produce because of insulin resistance, or more commonly, it's because the beta cells are sick or are dying because of the insulin resistance that I mentioned, or because of, in the case of type 1 diabetes, that there being a actual destruction and killing of the cells due to the immune system. So no matter what the origin is, the beta cell loss, diabetes is a result of there just not being enough healthy beta cells. And so the treatment that people are most familiar with is insulin injections in order to manage blood glucose levels. And this is definitely a life-saving therapy, but it's a very imperfect treatment. Essentially, the patients are trying to manually do the job that a beta cell would normally do inside of their body without them thinking about it. And in people and sensors and pumps and other electronic and mechanical ways of managing this are just not as good, nearly as good as the beta cell in terms of sensing sugar and secreting the exact amount of insulin that the body needs. And so that means that for many patients, control of blood glucose can be difficult, and that imprecision in controlling blood glucose is what leads to all the complications that patients encounter, particularly the reduced lifespan and increased risk of death. And so the idea that really motivates my laboratory is very simple on paper or on the slide here as a little cartoon, but in practice has been very difficult to achieve the way that we would all like, which is instead of relying on insulin injections, can we take a external source of islets containing insulin-producing beta cells and transplant these cells into patients that have now these exogenous cells and tissues replace the function lost in the patient's endogenous cells. And this would be a functional cure for the patient's diabetes. So this is something that is done to a limited extent using islets from deceased donors. And on the screen here, I have one example that was published by some colleagues of mine a few years ago that I think kind of illustrates the power of a cell therapy approach for type 1 diabetes in particular. That this patient was on insulin before the islet transplant. You can see on the graph there on the left that the blood glucose levels of this patient were just wild, very uncontrolled blood glucose, which that does happen in a good number of patients. But after receiving these islet transplantations and now having the exogenous cells controlling their blood glucose and the patient not needing to inject themselves with insulin anymore, you can see how flat and nice that line is, with the majority of the time the patient's blood glucose is within a normal acceptable level. And so kind of thinking a little bit broadly here, if we consider that worldwide there are several 100 million people with diabetes, now thinking about this from a bioengineering perspective, this says to me that we can't just rely on deceased donors to provide this material, and instead we need to have a large-scale sustainable source of replacement cells and tissues to even begin to treat any meaningful fraction of people with diabetes. So the focus of my laboratory is to use stem cells as this renewable source of cells. So in particular, using human pluripotent stem cells, either embryonic stem cells or induced pluripotent stem cells, or iPS cells, which can self-renewal to make a virtually unlimited number of themselves if given the correct cues, or given a different set of cues, can differentiate into any cell type found in the body, including the insulin-producing beta cell. So we published several publications at this point showing different ways and culture formats and scales for making these. But kind of no matter which iteration of the protocol you're talking about here, the end of this process produces clusters of cells of about 1,000 or 2,000 cells per cluster, for which the majority of the cells are capable of producing insulin. And so again, there's a lot of trainings in the call, so I've actually included several slides here that are a little bit more basic biology before we get into the details of the differentiation process and what we can do here. Like I mentioned, we use two primary sources of pluripotent stem cells, embryonic or iPS. The pluripotent stem cells there's markers that we look at and can look at to ensure that we have good quality cells. These cells, if given the right signals, mostly BFGF and TGF-beta signaling, are capable of proliferating and self-renewing virtually indefinitely. In my lab, we've done this for over 100 passages and can keep good quality on these cells. Or again, here's just some examples of their ability to differentiate into different lineages. Neurons is a picture I pulled from my colleague here at WashU, or heart muscle, which is a video from my PhD work actually. So the neurons and cardiomyocytes represent the ectodermal and the mesodermal germ layers respectively. So beta cells and a lot of your internal organs actually come from the endodermal germ layer, which has been historically just a very difficult germ layer to produce at all, let alone downstream differentiated and functional cells that could eventually be used for therapy. And so conceptually here, how would one take stem cells with the ability to self-renew and with pluripotency and figure out how to actually make these stem cell-derived beta cells? And there are many factors that people in the field can control and do control during differentiation that can influence what kind of cells you get from a differentiation process. And kind of early on in terms of my entry into the field, which occurred at about 2011, that the field had figured out what these signals were for making a high percentage of cells that were indeed endodermal in nature, and also were able to make cells that were pancreatic in nature. But subsequent differentiation with these pancreatic progenitor cells resulted in endocrine cells that indeed did have insulin. I mean, insulin is one of the big things that defines a beta cell, but were otherwise really not like a beta cell. They were dysfunctional, they were polyhormonal, and they had a lot of problems, which I have listed here on the slide. And just going to show you some data to kind of back this up. So again, this is some of the progenitor cells that people had made, and again, we have really good markers for characterizing and quantifying a differentiation process to make sure that we have a high quality differentiation going on to make high quality cells for therapy. But again, in the early days, before the field had figured out how to make beta cells, we can make cells that can produce C-peptide or insulin. So you'll see throughout the presentation that C-peptide is oftentimes what we're saying here. And that's really for technical reasons. And if people want to hear more about that, just ask me at the end of the presentation about that. So when you see C-peptide, it really means insulin in your mind. So here's a differentiation using these old protocols. You can see a lot of the cells are indeed producing C-peptide and insulin, but a lot of them also are producing other hormones like somatostatin and glucagon, which beta cells don't normally do, is other cell types in the islet that would normally produce these hormones. This is already a indication that these cells were not true beta cells. These cells also didn't have other markers besides insulin or C-peptide that you would expect a beta cell to have, like PDX1, which is one of the most important transcription factors for a beta cell to maintain its identity transcriptionally. And you can see here on the left, there's just basic polarization of the islet tissue, where one hemisphere has C-peptide and the other one has PDX1. But probably most importantly is that even though these cells could produce insulin and C-peptide, they were not glucose responsive. The number one functional feature of a beta cell is to secrete an elevated level of insulin when the cells are experiencing an elevated level of glucose. And we can measure that in the laboratory, and we found that these cells actually tended to go the opposite way. They would secrete lower amounts of insulin in response to high glucose, which is really much more like what an alpha cell, glucagon expressing alpha cell, would do, and not what a beta cell should be doing. In addition to that, the amount of insulin here was about 1,000-fold lower than what a real beta cell would do. So even though these cells didn't have the correct markers, they were just completely useless when it came to diabetes cell replacement therapy, because of the lack of proper regulated insulin secretion and just secreting way too low of levels of insulin in order to be therapeutically relevant. However, I did assert earlier that these pancreatic progenitor cells were correct. I don't have disclaimers here about them being dysfunctional or other kind of negative words to describe them. And that's because the field had figured out pretty early on that even though that these insulin expressing cells were dysfunctional, not useful for therapy, that these progenitor cells were indeed the correct type of progenitor cells. And the way the field figured this out was that if you transplant these cells into mice or rats, wait about half a year, then a fraction of these cells could spontaneously differentiate into these stem cell-derived beta cells, or I'll be calling them SE beta cells throughout the presentation here. And this is an example of that transplanting some pancreatic progenitor cells, waiting about half a year, you can get reasonable levels of C-peptide and also insulin detected in the serum of these mice, showing, again, spontaneous in vivo differentiation could occur with these cells. But of course, the big question was, how could we actually do that in vitro? We can't just go and inject cells into a mouse, wait half a year, scoop it out of the mouse, and put it back into a human. I don't think anybody with a CMC background would be very, very happy with that. Not to mention, regulatory and yields and throughput here. So we wanted to be able to do this in vitro, and we wanted to be able to do this not requiring 6 months of differentiation time. And so one of the things what we ended up doing here is we basically scoured PubMed, looked at basically every developmental biology paper that had been published, tested a lot of compounds, a lot of combinations of compounds in order to figure out which compounds were actually important in our differentiation process. And of course, the dirty little secret here is that most of the compounds reported in the dev bio literature just simply had no positive effect in our in vitro differentiation systems. This is very much a iterative, empirical slog through all these papers to eventually arrive at the differentiation process that is shown here in the schematic, where we take these pluripotent stem cells and we've broken down the protocol into 6 stages. Each of these essentially trying to mimic both in terms of the compounds given and the intermediate cell type produced here, trying to mimic the major steps that occur during normal embryonic development. But of course, we're doing this in the lab. We're not doing this in in vivo or embryonic environment. And so for each of these stages, there's different combinations of small molecules or compounds to push them into more and more of a fate, of a restricted fate, until finally we end up with a population for which about half the cells are these stem cell derived beta cells. And kind of the key thing here was really the understanding of needing these compounds in combination, particularly thyroid hormone gamma secretase inhibition and TGF-beta inhibition, was critical in order to get efficient differentiation here at stage 5 from PDX1, NKEX6-1 transcription factor expressing progenitor cells into an endocrine progenitor that still had competency to become a beta cell, rather than producing the polyhormonal dysfunctional cells I showed a few slides ago. And so there's a lot of data to back this up. I'm really just cherry picking this here for the sake of the amount of time that we had to speak about this. But here on the left, you can see a section of these islet aggregates that I showed several slides ago. Again, now here for these stem cell derived beta cells, seeing co-expression of a C-peptide with, in this case, we're just showing the beta cell marker NKEX6-1, like you would see with primary human beta cells. And again, the polyhormonal cells would have this segregation of a C-peptide from the other beta cell markers. So we're able to make cells that had most, but not all the markers of a beta cell. But probably most importantly, they were functional enough in order to be able to reverse diabetes in a mouse model. And so here this is a glucose tolerance test showing blood glucose levels after an injection of insulin. So these diabetic mice had received the polyhormonal cells, which is super high blood glucose levels, no matter what time point we looked at there, because these cells were just not able to be functional and produce enough insulin to control blood sugars in these mice. But you can see that our stem cell derived beta cells and our primary beta cells were able to clear out the injected glucose within an hour or two, which you would expect from a healthy human, or in this case, a healthy mouse. But you can see there was differences between the response from the mice receiving SE beta cells versus primary beta cells, and that's because even though we had made cells that were a lot better than the polyhormonal cells, they were still quite immature compared to primary beta cells from adult deceased donors, which I'll talk about a little bit in a few slides. But so we're very excited about this. We published this in Cell in 2014, and then this technology was transferred into industry, went through additional development there, of course, and is currently the subject of a clinical trial being run by Vertex Pharmaceuticals. They started this last year. They transplanted 3 patients so far, at least last that I heard. And this is a New York Times article on the first patient to receive these cells, and he has gone from needing, I think it was 34 units of insulin injection per day to no insulin anymore, because these artificial stem cell derived islet cells are able to control his blood sugar, provide him with enough insulin in order to no longer require insulin injections, which is something that we're very, very happy about. I think this is very promising for the future of the field of regenerative medicine. So when I formed my lab at Washington University, I had some immediate goals in mind that I wanted us to tackle when it came to these cells. Is that we really wanted to improve the generation of these cells and have an improved cell product, because I made reference before that while these cells were good and were functional and could have positive improvements in glycemic control in animals, and at the time we didn't know it would work in humans, and of course retrospectively we know that now, but there was definitely a lot of improvements that we perceive that we can achieve with these cells in terms of increasing the composition, improving the composition of the aggregates, and also improving the maturation of these cells. And then we also wanted to see if we could get any new biological insights from studying these cells. And so one of the things that we did was, again, another exercise of just empirically testing out different compounds or different media formulations, and was able to come up with an enhanced differentiation protocol, particularly when it came to stage 6, that would improve the function of these cells. A big part of that was really having a better control over the 3-dimensional assembly of these aggregates into aggregates that were just not like really huge. These were maybe closer to brain organoid size, which as opposed to like real islet size in the original protocol. And so we developed methodologies for controlling the assembly of these cells so that we have control over how large the aggregates are, and we have kind of a happy medium for which the cells are maximally functional, in addition to coming up with a enriched serum-free media that really helped with the maturation of these cells. So now again we get these nice clusters that are not too big, have the markers we'd expect of a islet. As again, this is just showing another panel of the usual suspects that you look at for either beta cells or for other cells that you'd expect to see in an islet, like alpha cells and delta cells, for which these are definitely present in our cellular drug product. And then in the end here, having a improved differentiation that is just more functional than the original protocol. And that's probably best shown here on the graph on the left, where we did a head-to-head comparison of the original 2014 protocol to our newer 2019 protocol, in benchmarking this to human islets. This is an in vitro functional assay where we just measure how much insulin is secreted by these cells over an hour, with the cells being either at low glucose or at high glucose. And you can see the 2019 protocol has great improvements in insulin secretion versus the 2014 protocol, but again, still not quite as good as what like the real thing is able to do in terms of primary islets isolated from deceased donors. A additional functional feature of these cells is it's not just the amount of insulin being secreted here that's improved, it's the kinetic release of these cells over time that is also greatly improved with these cells. So when you put islets into a perfusion system, so you can perfuse buffer over the islets, so you can control what the glucose concentration is in that buffer over time, and then like every minute or two collect the flow through there. That gives you a temporal measurement of shifts in insulin secretion of these cells. And so in this case here, I'm showing you data for which we were flowing low glucose over the cells, then step change to high glucose, and then step change back down to low glucose. And the human islet data here that you see with the upside down triangle shows you what is expected to be seen for a real functional islet, that within a few minutes of switching to high glucose, you'll get a sudden spike in insulin as a first phase response. With continued time at high glucose, you will have a lower than the peak, but higher than the low glucose baseline level of insulin secretion. It's called a second phase response. And then you would want, when the cells then go from high glucose to low glucose, you would expect there to be a tampering off of the insulin secretion back down to a normal baseline of insulin secretions. The human islets are showing that quite well here. Our 2014 protocol, the cells weren't able to do that. So it was actually one major functional feature that was missing with the cells made with the original differentiation protocol. But now here with the 2019 report, we have other protocols that are able to do this even better now. But we do have cells that do have both the first phase and second phase response. But again, not quite as good as the real thing. So we've also looked at the lifespan of this dynamic function during the last stage of the differentiation process. And we found that it took about a week of stage 6 in order for this functional feature to be observed. And it was able to last for 2 to 3 weeks afterwards, with the loss of function being mostly because of not the amount of insulin secretion at high glucose, but because the cells were losing the ability to keep a dampened amount of insulin secretion when the cells were at low glucose levels. So there is definitely a lifespan of optimal function here during in vitro culture. So once we'd established this new protocol with enhanced function, this presented a very obvious biological question to us that we didn't know the answer to, which is basically what is happening during the first week of functional maturation here. We knew that we had a media that was good at making the cells functional, but we didn't understand molecularly what was going on inside of the cell for them to be able to achieve this this acquisition of dynamic function during the first week of stage 6. And so we ended up doing a RNA sequencing experiment and identified the transcription factor 6-2, so this SIX2 is a very confusing name, that was most differentially expressed between the 2014 protocol and the 2019 protocol. So this is a transcription factor that had been associated previously with aging in human islets, but hadn't been studied in stem cell derived islets, let alone in more of a developmental context, which would be more relevant to our stem cell derived islets. Also, interestingly, this transcription factor is not expressed by rodent beta cells. So it was, in general, a very understudied gene that piqued our interest. And so when we went back to our differentiation protocol very simply looked to see when during the protocol was 6-2 actually expressed, and we found that most of the expression was gained during the actual last stage of the differentiation. And looking with finer timing here during stage 6, we saw a really nice correlation in the amount of secretion of these cells to the expression of 6-2. And looking at stage 6 cells and what cells were actually expressing 6-2, we found that it was only the endocrine cells that were expressing it, including the beta cells, but the alpha cells were not expressing it. We didn't see co-expression of glucagon and 6-2, for example, and it was not something being expressed by the progenitor cells. So we went and used CRISPR-Cas9 to create 2 knockout cell lines. We also did the same thing with lentiviruses with hairpin RNAs, but I'm just showing you the CRISPR data here. So with the homozygous knockout and a subsequent differentiation, we discovered that we were indeed still able to actually make the beta cells, as shown here with the C-peptide staining in the absence of 6-2. And these cells were still able to make the beta cell. They were still able to produce insulin. They were still able to secrete insulin, but the cells had absolutely no ability to respond to glucose. Again, now looking at this dynamic assay here, you see that the knockout cells with 6-2 knockout had a complete flat line, did not care at all what their glucose level was. It just secreted a constant level of insulin independent of what the glucose levels were. And that was also seen in the static assay. And so, again, I like to show this in my presentations because it shows how a focus on translational research can also open up new questions in biology, and the way that we my lab operates, that these go hand in hand, that we want new insights to biology to help with the translational work, and then advances the translational work to open up new biological questions that we end up focusing on. So now to shift gears here for a moment, I've been really focused on the differentiation protocol from very much a stem cell biology perspective, using small molecules and growth factors that pretty much only target signal transduction pathways within the cells. And I have that kind of shown in a schematic form on the slide here. But one of the things, especially if you think about this from more of a bioengineering perspective, one of the things that's missing from this is the understanding of what the contributions of the insoluble microenvironment would be on this process. And obviously, it's been really well studied in other systems, such as MSC differentiation to bone, cartilage, and fats, but it's been mostly not studied in the context of making pancreatic or making islet cells. The only real exception is that we did know that the assembly of the islets was important for the ultimate functional potential of the cells you made at the end of the process, but it still left a lot of the protocol being unexplored in terms of the role of the insoluble microenvironment in this process. And so one of the things that we did know before getting into this line of research that was kind of a mystery to us is that the differentiation process at the time required the cells to stay as aggregates during key stages of the differentiation process. And so if you took the protocols I showed earlier and you just repeated the protocols for the cells, instead of being in an aggregate, instead of being attached to the bottom of a plate, we were not able to be successful in making these islet cells. So it tells us at least very broadly that there was at least some sort of influence of the microenvironment on the differentiation, since the format was very key for the prior protocols to actually work. And so this led us to think that one of the big differences between the cells, again, very broad and somewhat superficially, one of the big differences between cells sitting in a three-dimensional aggregate versus being on top of a polystyrene plate is the rigidity or stiffness that the cells are experiencing, with the cells being quite squishy in a squishy 3D environment in pulling on each other, versus polystyrene, which is very stiff, several orders of magnitude stiffer than cells. So we ended up doing this very simple collagen gel height assay to try to understand if there was a relationship in making endocrine cells during differentiation with stiffness. And again, it was a very simple experiment to do, but this ended up being one of the kind of eureka moments of the lab that kind of opened up a whole new world of research and protocol optimization that my lab is still very actively working in today. Because again, simple experiment, but it showed very clearly that yes, there was a dependency on the cells become endocrine. So these are just 3 endocrine markers here. We had a lot of others that we looked at as well, but we were able to show here a clear connection of endocrine cell fate and stiffness. And thinking about that a little bit more, and kind of how the cells could sense stiffness and how that could potentially influence differentiation, we ended up curating a very small compound library, because we did that by using hydrogel. So while we could use hydrogels as a differentiation, we didn't think it was a very scalable way of doing this in a kind of a translational setting. And so we wanted to see if we could instead kind of take the insights we learned from that and translate it over to something that would be more scalable from a manufacturing perspective. And so small molecules is what we thought about there. We curated the library, tested it out, and we found that the actin fiber, the actin cytoskeletal polymerization inhibitor compound, the trunculin A, was a very potent inducer of endocrine cell fate. and these other ones didn't really work out too well. So again, so the triglycerin A depolymerizes the actin cell cytoskeleton. You can see that with the loss of polymerized actin here in the bottom right panel, and we quantified this with Western blot. And again, with triglycerin A, we were able to go in and confirm indeed that not only was actin being depolymerized, we were getting increases in the endocrine generation of these cells, either with immunostaining, or we confirmed this as well with this dosing study of different concentrations of the triglycerin A. It saw a strong dose-dependent relationship between the triglycerin A and endocrine gene expression. And so with that sort of understanding, and it was very unexpected at the time to see this, we wanted to understand it a little bit more. And so we did a single-cell RNA sequencing experiment where we took pancreatic progenitor cells and either treated them with nothing or treated them with a triglycerin A to depolymerize the cytoskeleton, or treat them with nocodazole, which depolymerizes microtubules, but that causes a hyperpolymerization of the actin cytoskeleton. And just ask in a fairly unbiased way, what was the cell fates that were that came out as a result of these treatments. And so here we were able to do that. We were able to identify retention of progenitor cells, some endocrine cells, some exocrine cells, and some kind of unknown progenitor cells. And then when we looked at what treatment group led to which cell fates, we found that the no treatment group was primarily made up of pancreatic progenitor cells. That the triglycerin A group was made up, majority were these endocrine cells, again, confirming what we had seen in the previous slides, but now going and hyperpolymerizing the actin cytoskeleton, we saw that the opposite cell fate was showing up instead, that we were instead of making endocrine cells, we were now making exocrine cells. And that led us to a understanding of that these progenitor cells essentially need a Goldilocks amount of actin cytoskeleton in order to stay as a progenitor cell. If you depolymerize that, you shift the cell fate from a progenitor fate into a endocrine fate. And if you hyperpolymerize, you shift the cell fate from a progenitor fate into a exocrine cell fate. And so now that's part of what we see here, that we really want to have actin cytoskeletal depolymerization to maximize induction to progenitor cells. But again, this still leaves a lot of other areas in the process for future study of the insoluble microenvironment. And so now when we take the protocol that either uses the sort of cytoskeletal modulation versus just using our 2019 suspension-based protocol, we saw something very, very interesting, is that in that some cell lines didn't care what protocol we used, and some cell lines cared a lot for what protocol that we used. And in general, cell lines that worked quite well with the suspension protocol also worked well with the cytoskeletal protocol. Again, we got comparable results there. But then other cell lines that didn't work as well with the suspension protocol actually worked quite well with the cytoskeletal modulation. And so basically the new protocols that use cytoskeletal modulation in conjunction with the targeting the cyclical transduction pathways that I mentioned earlier can basically help to improve cell line reproducibility and overall just make higher quality cells for study and for transplantation. And so now shifting gears just for a moment here to transplantation. So we took these cells either made, so we took cells made with the cytoskeletal modulation version of the protocol and transplanted into mice with severe pre-existing diabetes. And we did two cell lines here, an ES line and an IPS line that had been differentiated. And we saw that the reversal back to a non-diabetic state were comparable between the ES and IPS cells, but more importantly, we're also comparable to a equal amount of human islet cells also being transplanted here. So now we have cells that are able to reverse diabetes at a rate comparable to what real islets and real beta cells are able to do. And we're able to maintain this restoration of normal glycemia for a very long time. We show over half a year here, but we have gone for a whole year in my team. And then when you remove the graft, they're transplanted to the kidney, so we just remove the entire kidney. Then we have a reversal back to a diabetic state, showing indeed it is the graft that is causing this improvement in the blood glucose control of these mice. So again, now with the technical achievements and making cells that were even better than the previous protocols, this again opened up the biological question of what is actually happening during this few weeks it takes for the cells are actually able to fully control the mouse's diabetes. We do some things. There's probably some vascularization and wound healing going on there, but we really wanted to know, was there additional maturation events that were going on, and were the cells basically becoming better as a function of time in vivo? And we had some data already to suggest that to be true. We do that with extended time of transplantation, that the glucose for mice being transplanted with these stem cell derived islets. So we would have improvements in glucose tolerance. We would have increases in C-peptide or insulin secretion. And we knew that the cells were persistent for a long period of time. Again, this is just a picture from half a year. So the question was, do the cells mature after transplantation? And the way we chose to go and answer this question was using single-cell RNA sequencing. So we took cells that were transplanted into mice after 6 months, took the kidney out, manually essentially cut out the graft, dissociated it, ran single-cell RNA sequencing, and then bioinformatically removed any contaminating mouse cells in there to, in the end, get a population of cells that were just the human cells after transplantation to compare to the cells before they were actually transplanted. This is a very hard process that took us a long time to actually master and get to work reproducibly in my team. And so we were able to go and, kind of using standard markers of beta cells, we're able to identify all cell types we'd expect to see, and in particular the beta cells, which are shown in purple here. And then what I'm showing you here is just looking at the beta cells for either the in vitro stage 6 cells from the SC islets, or the beta cells from cadaveric human islets, or the grafted SC beta cells that have been in mice for 6 months. So you can see kind of broadly here that the grafted cells have a gene expression profile more similar to the human islets compared to the in vitro cells. Now we're making cells that have been in mice for 6 months that are looking a lot more like the real thing compared to the in vitro cells that have not been transplanted. When we look at the genes that change with time after transplantation, we see a bit less than 1,000 genes have changed expression from transplantation, and this includes increases in genes associated with maturation, such as insulin, IPP, and MAFA, and decreases in genes that are considered not desirable to have in a beta cell, such as FEV and somatostatin and glucagon. And I just want to highlight some of the maturation genes here, just separating this out a little bit more. So G6PC2, IPP, and MAFA are well-known maturation genes that in vitro stem cell-derived beta cells express at a low level compared to what primary human beta cells express. And that's shown here, that these green bars, green violin plots, are the primary beta cells. The next plot here is ES cells, differentiated into beta cells. You see very little expression of each of these 3 genes. But for these transplanted, we have 5 mice being shown here. But these 3 and these 2 mice now have expression of G6PC2, IPP, and MAFA at levels that are now comparable to the primary cells. So indeed, the C cells are undergoing changes in their identity to assume a more mature beta cell identity with time. And so I have maybe just a few more slides here that I want to get through. And this is just to kind of highlight some of the 2 main application areas that my lab and other people who are working in this field are looking at when it comes to stem cell derived beta cells. And the first one is cell transplantation. I've already showed you a lot of data associated with the function of these cells and the potency of these cells. There's a lot of complementary research that's going on out there. A lot of it dominated by people in bioengineering labs in order to really help with the translation of these cells into the clinic. And one of the things that my group had done early on in my time at WashU is to 3D print this kind of lobster cage to allow for us to potentially to have vascularization occurring, but also to be able to eventually retrieve the cells that were transplanted into the animals. So you can imagine that this requires a balancing of understanding the biology of the stem cells, the biology of the islets, how to 3D print cells. We did a lot of modeling of diffusional oxygen consumption and diffusion to make sure we're doing it in such a way that we weren't starving the cells being in this lobster cage, and also being able to successfully transplant and retrieve these cells. So that's one example, and it takes a lot of different disciplines to be able to address. The other one is encapsulation to provide these cells with immune protection. So this was a study that we published last year in collaboration with Millman Ma at Cornell University, for which we took the cells that we made and worked with the material that the Ma Lab had developed for encapsulation and immune protection, and we're able to show good success in immune competent animal models and also a dog model to not only have function of the cells, but also convey immune protection of the cells by use of this nanofiber technology that the Ma Lab had developed. So then the other area is in the area of disease modeling. And so most of the work that my lab has done there is to derive iPS cells from patients with diabetes, differentiate those into islet cells, and then study these in vitro derived patient-derived cells. And the first paper we published on this was with iPS cells that we had made from patients with type 1 diabetes. And this is a very interesting case because these are cells that are usually not available for study because the beta cells are mostly destroyed by the autoimmune attack in type 1 diabetes. And so now using this technology has opened up a new window of investigation into a cell type that's central to this disease, but is normally not available for study because it's destroyed over the course of this disease. And then finally, one of the more recent papers that we published on this is studying a rare form of diabetes that's called Wolfram syndrome. So these are patients that have pathogenic variants in the WFS1 gene, the Wolfram syndrome gene, and those variants cause diabetes in these patients, along with other ailments. We're focused on diabetes here. And so in this study, we were able to, again, derive iPS cells from these patients, use CRISPR-Cas9 to gene correct these cells, differentiate them into the stem cell-derived beta cells that either had the gene corrected or also had, in parallel, differentiations with the unedited SE beta cells. And we're actually able to then do a head-to-head comparison of these cells to see that indeed gene editing the WFS1 gene results in a restoration of insulin content in these cells, improved insulin processing, and also improved function of these cells. And we went on to transplant these into the mice to also show that these gene-edited patient-derived cells were able to restore euglycemia in a mouse model of diabetes. So I hope that those were just what your appetite a little bit about what are future areas of investigation and application of these cells, but both in terms of the clinical translation, but also the avenues of research that it opens up for people in the future. And then just maybe end with a very broad statement about the future impact here is that we think that this technology will ultimately result in greater accessibility of cell therapy for patients, and then ultimately having healthier patients that have lower risk of complications and don't have to live every day thinking about their disease and managing their disease. So that's my last slide. I just want to real quick acknowledge all my funding sources, my collaborators at WashU and beyond, and then the amazing team that we have here in the Millman Laboratory at WashU. So with that said, thank you for listening, and I'm happy to answer any questions that you have.
Thank you, Professor Millman, for this fantastic talk. Many questions came in. I could see one of them is I could see those derived beta-like clusters are working great. As they may be immunogenic, how is it that it will be transplanted to the recipients? Will that be an encapsulation device or approach that should opted for? Or how shall we proceed to host them into the recipient?
Yeah, so I mean, I think this question came in before I talked about the encapsulation. So some groups are looking at the encapsulation. The company that is doing the current clinical trial has a second trial planned with a encapsulation device that I wasn't involved in, so I don't really know anything about that besides they are doing a encapsulation device. I am working with a biotech company right now to test an alternative way of dealing with the immune system, which is gene editing of cells to make them hypoimmune so that the cells won't get rejected. So that's probably, so I would say the two main areas that are being looked at by myself and others are encapsulation and gene editing to make hypoimmune cells.
Thank you. Great talk. Can you talk about the survival of the grafts post-implantation? How much of the graft, if any, is lost post-implant? It's been shown that with islet transplantation, there's significant loss due to a number of factors like hypoxia.
Yeah, so within the first week or so of transplantation, in general, you're going to lose some islets. And we have, so hypoxia is one of them, but there are a few other factors as well. We have not gone in and looked very precisely at cell death and retention with transplantation. I can tell you that at least broadly, the transplanted cells are, a lot of them are still there and a lot of them last until for the remaining lifespan of the animal. And so while I'm sure we have some cell death, but the ones that at least survive the kind of acute cell death that occur after transplantation are able to stick around and live and function for, you know, the year and a half or so of the mouse's remaining lifespan.
Another question, on what basis the small molecules were selected and are they FDA approved? Are there any side effects caused by these small molecules?
Well, I mean, so these are not molecules that we would be injecting into patients. So there isn't really a concept of FDA approval of the molecules themselves. What I've been showing here is a research process. And so it hasn't been, so while others are doing this, and I can't really speak too much to that right now, but while others are looking at having a process that would meet quality and regulatory muster, that was not the focus of this research. And instead, we were using research grade reagents and not GMP or GLP grade reagents. With that said, most of these things are just small molecules for which sourcing to a GMP source is a fairly straightforward thing to do.
Thank you. Depletion for beta secretease or gamma secretease, is it possible in order to lower the risk for Alzheimer's disease?
I'm not sure that I understand the question. I mean, so we do use the gamma secretase inhibition, and that's a major signal we give the cells to promote their conversion from a progenitor cell into a endocrine cell, in addition to the actin depolymerization that we talked about there. I don't really know what the connection to Alzheimer's disease that's being asked about in the question, so I can't speak to that.
Okay. I think he's saying maybe this gamma can reduce the secretion levels in vivo, which may lower in the future Alzheimer's disease. But anyway, sometimes after transplantation, we do see decrease in this physiological glucose level until a certain period of time. But later again, it shoots up. When the grafts were explanted, we found them greatly vascularized and giving positive insulin staining. But why do we encounter something of this kind? Is it that the animal model has become insulin resistant?
Yeah, so I mean, it's always possible that the animals can become insulin resistant. So a lot of that might depend on the details of the experiment being asked about here. I personally haven't seen any evidence of insulin resistance resulting from transplantation of either cadaveric islets or stem cell derived islets. What I would suggest that the person who asked the question will look at there is when you have the improvements in blood glucose levels, are you having a appropriate or an expected level of human C-peptide or human insulin that correspond with that? And what I suspect is happening here, that while the blood glucose may have been improved, that does not correlate with there being sufficient amount of insulin or C-peptide to mean that was actually responsible for the blood glucose lowering. And so the blood glucose lowering could be occurring in that circumstance through a mechanism that is not because of the functional potency of the beta cell. And a few examples of that are if the mice are sick, they're not going to eat, their blood glucose levels will go down. That's one example. There's also other reports of being able to transplant like non-beta cells, such as brown fat, into animals and having improvements in blood glucose that's independent of restoration of insulin levels. And so while the blood glucose drop can be real, it could be in a way that's independent of the function of your beta cells, and therefore is not a good indication of translatability into a human.
Thank you. Regarding this implantation, are you implanting individual cells, or is there maybe make an organoid with a three-dimensional format? And also, how are they attaching? Are they becoming part of the pancreas, or are they in the blood vessel, or where?
Yeah, so we transplant them as aggregates of cells, about 1,000 or 2,000 cells per aggregate. That's a natural way the cells are within the pancreas, or in these like aggregates of cells. So we try to recapitulate that in the laboratory and inject them as in a solution, so the a loose solution of aggregates. For the work I showed here, we actually transplant into the kidney. It's just a convenient place to put it, and it's a very well vascularized location. Transplantation into the pancreas is hard. The pancreas is mostly digestive cells, so I don't really know how you would do that in a way that you wouldn't either cause pancreatitis in the recipient, that's one fear, or cause the digestive cell juices of the pancreas to eat up your islet cells. Clinically, it's usually done in the liver. We don't do that in mice just because it's really hard, because the mouse liver is like really small and I'm not really good at my hands to be able to inject it into there. But there are some more skilled surgeons than me who can actually do that.
Thank you. Another question is, how do you deal with scaling? How many aggregates, or how do you determine the number of aggregates to implant a patient?
The answer is empirically, unfortunately. So there's a lot of literature out there on transplantation of cadaveric islets into patients, and rule of thumb for that is that you want at least 10,000 islet equivalents per kilogram of the recipient for transplantation. That gives you kind of maybe an order of magnitude of what to aim for there. There's I would say controversy in the field in terms of how you would translate animal model data into to figure out dosing for a patient. Some people think you should be doing work in pigs. Some people think you should be doing work in non-human primates, but those models themselves also represent challenges in terms of the translatability over to a human, and I don't think there's a really good consensus on it.
Thank you. How is hypoimmunity controlled? Is it through HLA editing? And is the mechanism of hypoimmunity related to cellular response or humoral ones?
Yeah, so there are many different groups out there that have very different strategies. So I'm going to try to summarize very briefly because I see we're at time now. A lot of groups do delete HLA to prevent adaptive immune recognition. This also seems to be how the beta cells are killed in type 1 diabetes, is recognition of autoantigens by the autoimmune recognizing T cells there. So a lot of groups delete HLA. A lot of groups also overexpress markers on the surface of the cells to act as a don't eat me signal, particularly to block NK cell or macrophage killing. So like PD-L1 is a very common one. There's some people who do like CD47 is another one. And then there may be a third category here is deletion of like secreted factors such as prevent immune cells or even trafficking to where the beta cells are to attack them. Like I think Doug Melton a few weeks ago had a paper on CXCL10 deletion, I believe it was, to block that. So there's a lot of different ways out there. There's definitely not there is not a definitive best way of doing this right now, but a lot of groups are looking into it.
Excellent. Well, that's all the questions I have. Thank you for the fantastic talk, Professor Millman. Appreciate your time.
Great. I appreciate the invitation. Thank you.
Thank you. Bye-bye.
Bye-bye.